The significance of RNA replication. The evolution of life began with a single piece of RNA. Many people tend to get caught up in religious or ethical views when it comes to how life started, and people ignore the science that it is encompassed by. Chemistry, however, is arguably the most important factor in the evolution of life. Specifically, RNA replication is where it all began. RNA replication is so significant in the evolution of life because it is evidence of life in its most pure form. The pre-RNA world is the chemical process which formed RNA. Molecules started a little bit before the creation of RNA, and then they came to form as RNA molecules as they were discovered with phenotypes and genotypes. The pre-RNA world begins with the primitive atmosphere, which contained the building blocks of RNA, such as CO2 and N2. The RNA world began with hydrogen cyanide and hydrogen sulfide, which were probably deposited by a meteorite or impact or reactions in the atmosphere. These molecules existed even before RNA. RNA was not a prebiotic molecule, but the elements that build it up were. People thought ribosome C5H10O5 struggled with stability because ribosome nucleosides never formed. They thought the formation of RNA was extremely difficult because one of its most important features wasn't even able to be stable on Earth. Eventually, people trusted that development of the phenotype and genotype sparked the beginning of RNA replication. The genotype is the entirety of a human's genetic makeup unique to each individual. The phenotype, in contrast, is the makeup of the human's physical characteristics. The phenotype and genotype lived in the same polymer, and therefore, no protein or catalyst needed to be synthesized for them to function. The phenotype and genotype allowed for there to be some identification of physical and genetical characteristics as well. This can be related to the replication process, which focused on adenine, guanine, cytosine, and uracil of the pre-RNA world. These nucleotides paired with each other to make RNA replication happen. Adenine paired with uracil and cytosine paired with guanine. Discovering RNA replication was one of the most significant moments in the pre-RNA world because it developed a greater understanding as to why RNA was able to exist. The prebiotic synthesis of nucleotides was sparked by a reaction which would have occurred in water or when there was no solvent for the reaction. Synthesis was constructed because the atmosphere was developing from a primitive Earth to a more stable Earth. Methane and ammonia were showing up, and they pushed the synthesis of the nucleotide. The synthesis of sugars soon began. Sugars were one of the most essential molecules of life, and having them as a resource allowed for RNA to exist in its stable state. The actual synthesis of sugar can be very complex. However, many people have explained it in a little more simple of a manner, such as polymerization, which is the process during which monomers combine to form a polymer. People thought that many other types of synthesis, such as purine, also occur. Ribozyme-catalyzed reactions were very important in the origin of the RNA world. The most important ribozyme was the ribosome, which starts the peptide bond formation in protein synthesis. These ribosomal conclusions as origins of the RNA world came from the molecular biologist stream, which concerned itself with the double-stranded structure of RNA, which functioned for RNA interference. The basic question was, how did it stay alive? Some of this was explained through molecular replication. Nucleic acid replication took part in the model of replication of a double-stranded helix for RNA. There was a three-step general process that took place. The first part is the separation of strands. The second part is where the dimer forms. The third part is where the dimer is acting as a primer to add another monomer. And the fourth part is showing that this process will go on until the template is full. Essentially, the double helix breaks up and the two strands replicate separately. During replication, mutations are caused by copying errors. High mutation rates indicate an inaccurate copying method. However, the copying of nucleic acids generally has very few errors. To eliminate as much error as possible, alternative models were consistently being looked at the time.
RNA was a much simpler protein than DNA. However, there was quite a messy path to discovering how RNA functioned. One of the most popular theories is the Darwinian revolution, of which before this, scientists focused on the ability of molecules to replicate, thus making the beginning of life. Prebiotic molecules outdated the Darwinian theory, which was based off of the assumption that natural selection and mutation of gene replication is how humans evolved. The assumption was that polynucleotides began to replicate and form RNA. Ribose, phosphate, and purines and pyrimidines were the building blocks for RNA. However, there was a messy path to their discovery. Nucleotide analogs were one significant roadblock in the pairing of the molecules above, which made the discovery of RNA more complex. These nucleotide analogs combined to form polymers. However, this got complicated when considering all the proteins and sugars that made up these polymers. Understanding these structures started turning questions of how other aspects of RNA functioned. In 2009, Sutherland discovered how to make complete nucleotides. Sutherland discovered cyanosulfidic chemistry, which explained how the building blocks of RNA formed. Sutherland formed the nucleotides by combining hydrogen sulfide and hydrogen cyanide in an aqueous solution to form a 3-carbon sugar, glyceraldehyde. Many reactions take place during this time, and the process is repeated. Sutherland also formed fatty acids from these 3-carbon sugars by using UV light to form glycerol and acetone from dihydroxyacetone. Sutherland then used acetone to form amino acid precursors. Sutherland believed that there were similarities between his discoveries and existing modern cells. This might indicate that RNA was the origin of life. RNA is arguably the most elementary level of life because of the proteins and nucleotides that build it up. These molecules came to be before any other molecules of life and bonded through chemical reactions to form RNA and its polymers. Theories like that of Charles Darwin and other scientists, which regard themselves with ethical and humanistic ape-type views, aren't credible because they don't break down to the simplest forms of organisms possible, like RNA. Sutherland's cyanosulfidic chemistry, however, revolutionized the hypotheses related to RNA replication. In the end, however, a lot of this theory is unproven, and that is why extensive research is still being done today.